Evolving pathogens are a major risk to public health, but understanding how pathogens adapt to new hosts remains a challenge. Some bacteria can transfer to new host species, and this poses a risk to human health. An estimated 60% of all human pathogens have originated from other animal species. The bacterial population from multiple hosts such as plant, animal, and human can be identified through the advances in sequencing technologies. These technologies have allowed the comparative genomic analysis, which provide new understandings into the evolutionary process of bacteria in new host species. Back to 2017, an experiment done by Murray and his team shows the recombination-mediated host adaptation by Staphylococcus aureus. This bacterium is a major cause of animal diseases including scale infections of poultry, which are a large economic problem on the global chicken industry. The emergence of poultry-associated lineages has involved zoonotic transmission, which simply means the disease is directly transmitted from animals to humans. The experiment is done by characterizing genetic variation in a population of genome sequences of this bacteria inside the isolates poultry and human. The result shows that poultry and human CC5 isolates were significantly different from each other, and more recombination events were detected in the poultry isolates. Recombination suggests that the combination of the genes on the genome may change due to such DNA rearrangements. In a population, this sort of genetic variation is important to allow organisms to evolve in response to a changing environment. Therefore, by combining phenotype information with evolutionary analysis of the bacteria genomes, the experiment has provided evidence of adaptation following a human-to-poultry host transition. The effect of the usage of antibiotics in animal feed have been well studied in relation to the evolution of pathogen populations in poultry. The major use of antibiotics in animal feed is to increase the productivity of the animals. After animals have been fed antibiotics over a period of time, they retain the strains of bacteria which are resistant to antibiotics. Through interaction, the resistant bacteria are transmitted to the other animals, thus forming a colonization of antibiotic-resistant bacteria. Transmission occurs when the bacteria is ingested. As an example, humans can get infected by eating meat from animals with resistant bacteria. Even though cooking reduces the survival of the bacteria, some may still survive and infect the human. Whole genome sequencing has opened the way for investigating the dynamics and genomic evolution of bacterial pathogens during the colonization and infection of humans. Recent improvements in sequencing technologies and analysis tools have rapidly increased the output and analysis speed, as well as reduced the overall cost of whole genome sequencing. From the experiment conducted, YGS improved the identification of the cultivated bacteria, and almost complete agreement was observed between the phenotypic and predicted antimicrobial. Now, YGS is becoming available as a routine tool for clinical microbiology. There are three main points to conclude this whole video. The first point is by reducing the amount of antibiotics used for animal feed to prevent resistance. The next point is to prevent the transmission of bacterial pathogens into our system, and one of the examples that we can do is by cooking our meat properly. Third point is the more research should be done to get a better understanding of the genetic and systematic basic for the bacterial host adaptation. By doing this, it may result in more solutions in controlling infection of new pathogens as well as to limit the emergence of new pathogens.